glycosylation has an important role in immune modulation, and in fact, most of the immune molecules and microbial patterns recognized are glycans or glycoconjugates. And sialic acids that are decreased in GNE myopathy are at the outermost end of glycans, and therefore they are really important for immune modulation. They can prevent the recognition of the underlying glycans by other proteins like galactines. They can also activate the complement pathway or regulate the cell immune response by being binding targets of sialics. Some previous work from our group also showed that the sialic acid content can modulate the cell surface stability and expression of the MHC class I complex. And this molecule is expressed by almost all nucleated cells, and it functions as a self-mark for our cells. And it's important for antigen presentation, for T-cell lymphocytes, and therefore for a correct immune response. So despite all this knowledge about the importance of sialic acid in the immune function, it is surprising that there is no correlation between GNE myopathy and immune changes or alterations. So with the previous work of our lab in mind, we decided to evaluate if MHC class I expression is altered in a GNE myopathy cell model. So we used the HEC293 GNE knockout cell model that was kindly provided by the MLU team, and this model was created by CRISPR editing. So it has the GNE gene knockout, and therefore the GNE protein is not synthesized. And in this model, we observed higher total and cell surface expression of MHC class I molecule. Okay. Oh. Then we decided to evaluate if sialation increasing therapies such as the progeny prodrugs or MANAC were able to modulate MHC class I levels. And first, we showed that the progeny prodrugs and the MANAC were able to restore the sialic acid levels in this model. And if you want to know more about this, there is a poster outside. Those results will not be depicted in this presentation. But then we thought that if there is a correlation between the sialic acid content and the MHC class I expression, we will notice a decrease of the MHC class I levels in the GNE knockout cell after supplementation. And in fact, we observed that with the concentrations of the products tested and also with MANAC. So then we wanted to know if our observations have any clinical value, because we are using a human embryonic kidney cell model. So we decided to evaluate muscle biopsies of patients. This is very important because there are very few reports of inflammation and MHC class I expression in GNE myopathy. So we evaluate muscle biopsies, and we can see that MHC class I staining, here depicted in green, is increased in the GNE myopathy patients compared to the control. So this is important because we can't dismiss GNE myopathy diagnosed when we see a markedly increased staining in the MHC class I. So conclusion, so far we observed higher MHC class I expression in a GNE myopathy cell model and also in muscle biopsies. And we also show that this could be modulated by the sialic acid content using the prodrugs and MANAC. Having said that, the mechanism by which the sialic acid content is regulating the MHC class I expression is unknown. It remains unclear, and it will be important to study how hyposialylation can be modulating the MHC class I turnover or the cell surface stability. And one hypothesis is that hyposialylation is enabling the recognition of the underlying glycans by other stabilizer proteins at the cell surface. So in the next steps, we would like to understand if the cell surface stability is higher in the GNE knockout cells, and if this could be to galactine binding by testing galactine inhibitors. Then we would like also to know if higher MHC class I presentation is correlated with higher antigen presentation, because we know that our model is not the best as an immune model, but we already evaluated an inflammatory cytokine that was IL-6, and our results do not support diseases of an inflammatory response, but we can discard the hypothesis of immune modulation. So we'd like to know if the MHC class I expressed at the cell surface is functional and if it can do antigen presentation. And to finish, we'll also like to understand which are the cells that are expressing MHC class 1 at the muscle environment, because typically muscles do not express these markers. So if they are immune infiltrates or fibroblasts, so we will evaluate patient-derived fibroblasts to know if the levels of MHC class 1 are also increased in these cells. So to finish, I would like to thank to all the glycoimmunology group and also to all the labs involved in this work.